We have to think that most of the rare diseases used to be multifactorial, multisystemic and multi-organic disorders involving several organs, maybe in many cases also with a major focus in one organ of the body, but used to be multisystemic. Even in that situation, again, most of them, between 70–80% of them, are genetic diseases and primarily caused by a gene variant, pathogenic gene variant in a specific gene, or several different genes, but in any patients, in each case, patient and family, the disease is really caused by a specific gene. Well, in this case, in the last 10–15 years, the number of diseases and disorders has been recognized because the gene has been isolated, has been identified, and is doing using the information for diagnosis, but also for research to understand the pathogenic mechanisms, and also looking for druggable targets, has increased a lot. So the speed of the recognition of new rare genetic diseases has increased in an almost exponential point of view. So we understand better not only the genetic basis of the classical, well-recognized Mendelian and mitochondrial diseases, but also new diseases that has been recognized from the syndromic point of view with not any etiology, and now we recognize the etiology that is genetic etiology caused by one specific gene or a number of genes related to that phenotype. So things has changed a lot, and obviously this represents new opportunities for diagnosis. In this case, we have to think that in the last 15 years, the ability of next generation sequencing, second and even third tiers of gene sequencing, because after the Sanger sequencing that has been the only way for a long time, now we have a lot of technology doing exome and genome sequencing has increased a lot. But not only the possibility to use the technologies to diagnose or to sequencing the exome and mainly the whole genome, but also the possibility to sequence the RNA, doing RNA sequencing, also to investigate episignatures. And from the genomic point of view, traditionally we have been working with short reads, and now we have the possibility to perform long reads. So we have now a good chance to find any single nucleotide variant or structural variants by genome sequencing in the patient's genome. So we can find one specific point mutation, but also we can in a gene, between a gene, or we can also recognize rearrangement, chromosomal rearrangements like microdeletions, microduplication, insertion, and so on. But the point is not only the next generation sequencing, it's also the possibility to do improving the quality of the structural variant analysis, doing obviously microchromosomal arrays or chromosomal microarrays, CMA, but also the optical mapping, that is another possibility to investigate the rearrangements regarding to the micro and doing deletions and micro duplications. This has happened for many patients and many situations. One of the things that I can remember not directly with patients when we found one of the genes related to Charcot-Marie-Tooth disease, that is a genetic neuropathy, one of the most common rare diseases group. How we found many years ago the gene encoding GDAP1 as a new protein related to mitochondria, and how this is generating different phenotypes related to Charcot-Marie-Tooth disease in both processes, severe forms, but also dominant milder forms of the disease. This has been impressive for me. And also, with thinking about the specific patients, my impression has been the possibility to think about one specific patient that you find a genetic variant that could be the cause of the disease, but is not very well defined because maybe the variant is of unknown significance and even in some cases, likely benign variant. But if the phenotype agrees pretty well with the found gene, it's also the possibility to work and to investigate doing functional genomic studies, trying to solve from the within silico and experimental cell and molecular biology tools, try to demonstrate that there is an effect from the biological point of view and pathogenic effect in these biological experiments. And this is nice because it's a way to put together clinical medicine that has to be always related to the very well-defined knowledge with experimental biology and putting together even inside intramural work in a hospital, in between the hospital and the close research institute to solve diagnosis of patients. So this has been very, very interesting for us. Is I have the experience working with a patient that was 80 years old for a long time. He was undiagnosed, and he had a important developmental delay. There was complete absence of language, and there was also another kind of problems. There was a couple of times that there was seizures, problems with regarding to the esophageal problems, and I found two variants in this gene, GMB5. One was pathogenic, but the other was really likely benign. And we could demonstrate that these variants were doing important changes in the biology of the gene, and then we can demonstrate that it was the cause of the disease. Well, I'm a medical doctor by training, and also I did my specialty in pediatrics, but there has been always interest in genetics, especially in human genetics. I like it. I like genetics as a biological and scientific discipline, and also the application in human genetics as a medical or clinical genetics approaches, and also including genetic counseling. So when I was in my third year of pediatrics, I decided to be in touch with the genetics unit to learn about clinical genetics and also to work in the laboratory to understand how could be the application of genetic for diagnosis, especially rare diseases that used to be monogenic disorders. So I was very happy. I was started with doing cytogenetics in leukemia, but also I moved to molecular genetics. I'm talking about the times of the 18th, and the time that the position of cloning was the way to looking for and to find new genes. And has been always very, very interesting because from genetics you can support diagnosis, you can looking for research on pathophysiology of the disease, also to looking for new druggable treatments, but also something that is very, very important from my point of view, that is genetic counseling. Altogether makes genetics and medical and human genetics very, very important in medicine from my point of view, and also feel that I'm really happy to be part of this field and to enjoy every day doing clinical work and now mainly research work on these diseases. If we are talking about young scientists, I think the career in genetics is very, very nice, specifically in human genetics, because you can have an approach, scientific approach, that's something very, very important in biology, of human biology. That's to know what is the basis of the inheritance, which is the basis also to understand why everybody is different to each other, even if within the same family, not only because the genetics of the inheritance, but also the interaction of the genome with the environment, and also how is the regulation of the gene expression, all the regulation, including the epigenetics regulation, and so on. This is very fascinating field for young scientists that would like to move in the field of biomedical sciences. But also, if we are talking about rare diseases, we have two things important. The clinical application of everything you are doing, specifically now mainly in diagnosis, but also in the future, well, in the present now, but also in the future, regarding to new treatments, researching treatments, looking for druggable or molecular targets. It's really very impressive how it's changing, and it's going to change a lot. But also in the field of rare disease, there is another point that is really very interesting. If you investigate the biology, genetics, the pathophysiology, and the clinical features in individuals, in patients living with a rare disease, you have also the opportunity not only to support these patients and families, but also to know better specific mechanisms and pathways that could be related to common diseases and therapies for common diseases. Because at the end, a rare genetic disease is the end of a phenotype that could be the expression of specific, very pregnant mutation, but could be related to another genes, to another pathway that could have susceptibility or generate susceptibility to suffer common diseases. It's like you have to understand what really happened many years ago regarding to arteriosclerosis, hyperlipidemias, and also the consequences of these hyperlipidemias and arteriosclerosis. I mean, and also new treatments. What really happened with the knowledge about cholesterol physiology and also statins as a drugs was started when Brown and Goldstein investigated and they discovered the receptor of LDL lipoproteins that are related when mutated in patients generating familial hypercholesterolemia, and now was the basis to understand better the cholesterol biology and also looking for new drugs that could reduce the amount of cholesterol and to prevent the development of arteriosclerosis. This is a way to understand what I mean when they're talking about the interest or the relevance of investigating rare diseases for common diseases. At the same time, everything that is doing in rare diseases is useful for the patients with rare diseases.